Thanks for the introduction, Corrine. I see many familiar faces, so thanks for coming out here today. As Corrine mentioned, I'm going to be talking about soybeans. I'll also be talking about 4 different trials. So I'm going to refer to that book of knowledge, as Corrine calls it, multiple times, and I'll be referring you back to the page numbers if you want more details. The first set of trials I'm going to be talking about actually provide a pretty good opportunity for us, one we don't typically have. It's all about Phytophthora soji. That's a pathogen of soybeans. And this is work coming out of the McCahey lab. Dean Malvik's involved, Jane Newbart here is involved, and then Kat Markham is involved as well. She's kind of heading up the project. But the overall goal of this project is to figure out the composition of the Phytophthora soji population in our soybean fields. For a bit of a reminder, Phytophthora soji can cause disease at any point in the soybeans growing season. So early on, you can see either pre or post-emergence damping off. So this is basically seedling rot. So you can see big gaps in the rows oftentimes. But later on in the season, you'll sometimes see something that's called Phytophthora root and stem rot. And this is characterized by this kind of chocolate brown lesion that's extending from below ground up and onto the stem tissue. Phytophthora soji, it's actually a sort of like a fungus, but it's not a fungus. We typically call these water molds or oomycetes. The reason why we call them water molds is because they produce swimming spores. So as you might imagine, this particular pathogen survives and thrives under saturated soil conditions. So clay soils, poorly drained soils, compacted soils, all of those are going to increase your risk for Phytophthora diseases. So here's a nice little cartoon showing what happens below ground when a plant is infected or being infected by Phytophthora soji. So starting over here on the left-hand side, these little pink blobs, those are spores of Phytophthora. They swim towards the root, and when they actually make their way to the root, they secrete something called an AVR molecule. Bear with me, this is kind of complicated, but I think it'll make sense with these cartoons. Pictures always help me. So these AVR proteins make it so that the pathogen can go in undetected. It sort of shuts down parts of the soybean's immune system so that disease occurs. Now, you might recall when you've dealt with Phytophthora soji and Phytophthora root and stem rot in the past, that most of the decisions that you're going to make happen before any seed has been sown. So we have 2 main ways of managing Phytophthora. One is by planting resistant varieties, and the other is through specific seed treatment, fungicides. So variety selection is key. So soybean plants have these RPS genes. RPS stands for resistance to Phytophthora soji. So these RPS genes have specific number and letter combinations. So this particular soybean plant in our cartoon has the RPS 1K gene. What happens is if we have in our field a Phytophthora isolate that carries this AVR1K protein, what happens is when it goes to try and infect the plant is able to detect it and mount an immune response. And so that's sort of how resistance works. Unfortunately, though, over time, With repeated cycles of infection, there's a lot of spores that are produced, and just through random mutations, sometimes you can have changes in our our AVR genes. So if, for example, you plant the same soybean variety with RPS1K, with that resistance gene, and your Phytophthora isolate produces an AVR protein 3A, you're going to get disease. And so over time, this is how populations shift. I sort of tend to think of it as an arms race. So with each resistance gene we're deploying in the field, eventually the Phytophthora population is going to evolve to be able to resist it. And so the overall goal of this project is to figure out which AVR proteins are around in our soybean fields. There's a couple reasons for this. One, we need our soybean breeders to be aware these are the RPS genes we need bred into our varieties. And then we need to be able to deploy the correct varieties adapted to our maturity groups up in the Northwest up here. So in the past, the way that this used to work, figuring out and doing one of these large pathogen surveys, is a lab would go out into a field, collect soil, collect plants, diseased plant samples, bring it back to the lab, isolate the fungus, and then use that fungus to inoculate tons of plants. So they have soybean lines with known individual RPS genes in them, and then they use that pathogen to challenge those varieties, and that's how they would figure out which AVR genes were present in the pathogen population. Now that takes a lot of time and money, and that's because for each line you need 10 plants and somebody to plant, inoculate, tend, rate, do everything that's needed for those plants, and in order to figure out which AVR genes are present. And there could still be false positives and negatives. So more recently, there's been developed a molecular assay. So they still can collect soil samples and plant samples from the field and isolate the pathogen, but then they use molecular techniques, and with 98% accuracy, they're able to figure out individual AVR proteins that are in our Phytophthora populations. So this past year, so here's a couple samples I've sent in from the region. This past year, they're collecting soil and plant samples. If you see something that looks like Phytophthora root and stem rot in your field, this is a really great opportunity because we can collect that material, send it into their lab, and they'll tell you which RPS genes are most likely to be effective in your particular field, given your pathogen population. So either contact me or send an email to Kat or Megan, and we can have somebody come in and collect samples for you. Okay, on to the next trial. So this is on page 32 of the Book of Knowledge. This is a white mold project. This again is coming out of the McCahey lab, and this is a particular graduate student, Alicia Hirschman. So she's doing a lot of the work for this project. So unfortunately, those of us in the Northwest are no stranger to white mold. This is sort of what it looks like in the field. And the reason why we're no strangers is because a lot of our plants, our broadleaf crops up in the Northwest, are susceptible to the pathogen. So the pathogen has a very wide host range, and it produces very long-lived survival structures that make it so that we're not going to rotate our way out of a white mold problem. Chemical management requires precise timing and really good coverage. And in a year that favors disease, we're still going to end up with some yield loss. We do have resistance available in some of our varieties. However, it's nowhere near complete. And we could do better. And so the goal of this study was to look at two different things with the hopes of developing a better resistance package within a soybean variety. So it's thought that plant architecture traits can contribute to the plants being able to escape infection by the white mold pathogen. So thinking about a soybean plant with a more upright architecture, the rows are going to be a little bit slower to close. There might be a little bit more light penetration down to the soil. You might end up with a little bit less of a humid environment under there. So it's thought that some of these plant architecture traits, if they're combined with actual resistance within the plant, can provide a better overall resistance. In addition to architecture, we also have something that's called physiological resistance. So this is when you actually challenge the plant with the pathogen, and the plant is able to genetically resist the pathogen. So you would have both the architecture and physiological resistance all within the same plant. So what they did is they looked at 150 different soybean lines, and then they narrowed that down to 20 different lines. And those 20 lines are all listed out here. They all have different colors, and each of these graphs, all those colors are represented. And they looked at a lot of different architecture traits, trying to develop a set of lines, so these 20 lines, that vary from low, medium, or high canopy closure at flowering, for example, low, medium, or high petiole slope. So if you remember, with petioles, those are those little, like the leaf stems that attach the leaf blade to the main stem. If you have a very low slope, so the leaf is standing more outward like this as opposed to this, that's going to create a more rounded canopy and probably not provide as much escape. So they looked at petiole slope, leaflet length, and width ratio. That's basically leaf shape. They also looked at leaflet area and then UVB light penetration at different distances from the stems. They looked at all different things. They also looked at disease in the field. So they wanted to see in a disease nursery, what happens? Were they able to, are they seeing evidence of this avoidance? And so what they had, so this table right here down at the bottom, they planted all of these soybean lines out and then had the inoculum in the field and then just waited for disease to occur. And then they looked at disease incidence, so that's what percentage of the plants have disease, and then severity, how bad was that disease. And they combine that into something called a disease severity index. And so any of these different lines here with a 0, that means that there was no disease. Anything with a value above 0 means that there was disease. They also had a couple of checks. So they had 2 different Asgrow varieties, one that has that more upright stature, and one that has a more bushy stature. So they did see disease in their bushy stature check, but not in the upright stature check. So what they're doing now is They're testing for physiological resistance in the greenhouse, because it would be ideal if they could find physiological resistance in some of those varieties that also have some of those plant architecture traits that allow the plant to escape infection. So they're going to repeat their field trials in 2024 as well. Here's just a result of our Western Minnesota IPM survey. This is in your Book of Knowledge, page 37. So quite a few folks from Minnesota Extension. We had a couple of summer scouts working with us, and then NDSU, the NDSU IPM program with Jan Knodel and Patrick were also involved with some of the mapping. So whenever a scout would visit a field, they looked at a bunch of different things within each field. So first thing they did is, upon approaching the field, is they used a sweep net to go in and sweep for grasshopper nymphs and adult grasshoppers in that grassy edge along the field. They also looked for these bean leaf beetles, and then the injury, the chewing injury that they cause. They looked at two-spotted spider mites, both at the field edge, so in the soybeans growing right along the grassy area, and then inside the field as well. So here's what they found regarding grasshopper nymphs, and this was from June 5th through 9th. All of those little black dots here, those locations were visited and they didn't find any nymphs. All the little green triangles, they found between 1 and 25 grasshopper nymphs. So if you were going to be treating your field based on grasshopper populations, have we yet reached the treatment threshold? You guys remember? Now, treatment thresholds are devised so that we can take all of the emotion out of our decision. You reach a treatment threshold, and that's where research has shown that if you don't treat at that point, you're likely to experience yield loss to your crop. This is about, even if you had 25, that's half of our treatment threshold. So when it comes to nymphs, outside the field, we need 50 or more. The adults outside the field, that's 20 or more adults, and that's per square yard. Within the field, that number drops. So 30 plus nymphs within the field, 8 plus adults within the field. So that's, if you're looking at population densities to figure out whether your treatment threshold has been reached, we didn't reach treatment thresholds, at least in those fields during that particular week. There's also another way to make a treatment decision based on thresholds, and that's particularly useful when you have multiple defoliators present. And that's based on looking at defoliation injury. So oftentimes you'll have more defoliation taking place towards the top of the plant. And so when we're talking about defoliation injury, we're talking about average defoliation throughout the entire canopy. So we would suggest taking a leaflet from the lower canopy, the middle, and the upper canopy, and then doing that at multiple locations throughout the field and figuring out an average of your defoliation injury. Luckily, we now have tools like this. If you just wanted to Google Crop Protection Network soybean defoliation training quiz, you can actually train your eye to be able to tell 30% injury from 40%, et cetera. So the treatment threshold is 30% injury on average throughout the field during vegetative growth stages and 20% during reproductive growth stages. Couple other things that were looked at in these fields: soybean growth stage, soybean aphid incidence and severity. So how many people had soybean aphids this year in their field? Threshold level? Some folks? Yeah. Yeah, that's what I saw too. So, after the last couple years, I was sort of hoping that aphids were kind of going the way they've gone in Illinois and Wisconsin. But yeah, they were kind of back with a vengeance in some of our fields up here. So in addition to looking at soybean aphid percentage of plants infested, they also looked at how many soybean aphids there were per plant. And they also looked for the presence of evidence of our natural enemies. So here, these are called aphid mummies. And these aphids have been colonized by a parasitized or a parasitic wasp. And so they're dead, and at some point another wasp is going to hatch out of them. But, our natural enemies do a good job for us some years. And if we hold off until treatment threshold, sometimes, we might get a nice rain that helps some of the fungi come in that can parasitize our soybean aphids. Those, along with the insect predators, can sometimes mean the difference between having to make an application and not. So here's soybean aphid severity. So this is July 17th through the 28th. So the dark red triangles, so there's a couple down in Mahnomen, there's one in Clay, and I visited those couple fields. That's soybean aphids, so 200 plus soybean aphids per plant. Is that the threshold, the treatment threshold? I see a couple of folks shaking their head no. Right. So the treatment threshold hasn't changed. It's 250 aphids per plant on average on more than 80% of the plants, and that population density is growing over time. So in these couple fields, we'll admit to going back and visiting those fields multiple weeks in a row. Where it lied, when I stopped looking, there were more than 1,300 aphids per plant in some of these fields. So I was hoping to not have to experience, the green pants, sticky pants, but unfortunately we did have some problems last year with soybean aphid. A couple of other things we had scouts look at were looking for some of the newest pests to make their way to Minnesota. One is this soybean gall midge. That was first found in the southwestern part of the state, and it's been moving north since. We didn't add any additional counties to our map, so our scouts didn't find soybean gall midge this year. One thing that we did find, unfortunately, is something called soybean tentiform leaf miner. So if you're kind of like me and you get into a field, you're just kind of trained to keep flipping those leaves over and looking at the underside, because that's where you're going to see all kinds of unhappiness. When you come across something like this really catches your attention because it's not like anything I had ever seen on a soybean plant before. So this is a new pest. It was found for the first time in 2021 in the southeastern part of the state. It does parasitize two different natural kind of American hog peanut, and I can't remember the name of the other kind of wild plant that grows around, but it's decided soybeans are also on its menu. And so if you looked at a plant that's infested from the top, a leaf, you might, if you look very closely, notice that something's going on there. So as the area of the leaf that that's mined increases, what happens is it kind of gets this tented structure, so it actually has relief. So you can actually see here, if you look closely, there's a bit of relief there, so it can kind of pop up. But still, that would be really hard to see unless you flip it over. So it's thought that these areas, as they get more mined out, it's sort of like defoliation injury. So there's a lot of work being done on this, mostly around St. Paul and in the southeastern part of the state. But I took these pictures in either western or eastern Marshall County, and it was only a couple years ago that it was just in the south. So it's here. Where I did happen to find it was right next to trees. Not like a field windbreak, but a large wooded tract of land. So if you're growing soybeans next to woods, wouldn't hurt to just go and start flipping over a couple of leaves and let us know if you find it. Because if it's here, maybe we can get some research up here looking at it in more detail. Did you guys see this guy this year? Yeah? Yeah, so this is the head. This guy has 3 sets of true legs. It has 3 sets of what are called pro legs. And then it has 1 set at the back. So what is this guy? Green clover worm, right? Yes. So I do have a handout in the back. It's ID and management of foliar feeding caterpillars. So please go ahead and take that. This can complicate matters, and I have a whole section about what to think about when you're trying to manage for this. So some people were able to hold off, but this guy right here, you'd use that defoliation, those defoliation estimates to determine whether you've reached treatment threshold or not. So what about chlorpyrifos? So this is something that's been in the news lately. So if you'll recall, that's the organophosphate active ingredient that's in, for example, Lorsban. So in just this past year, there was a U.S. Circuit Court of Appeals that decided, you know what, EPA, nope, we're going to set, basically, they tossed out the 2021 EPA ruling banning the use of chlorpyrifos, and the court said, you guys need to go through the process again of figuring out whether it's safe to use in food and feed crops. So, for those of us that were hoping that it would be back on the menu of products for us to use this coming year, unfortunately, I spoke with a scientist down in St. Paul that works for MDA. It's still, because it's not registered in the state, needs to go through the EPA, like the federal registration process first, and then the manufacturer needs to appeal to the state and say, hey, would you mind registering this for use in the state of Minnesota? So there's a couple steps that we need to pass through in order to, again, be able to use it. So at this point, no use, no sales, no distribution are allowed. One last thing, because my hair seems to be on fire about this, whether or not anybody else's hair is on fire. It's SCN sampling program. If you'll recall, SCN is that little worm that parasitizes soybean stem or roots underneath the soil. More information, it's kind of outdated, is in your book on page 27. So we had a sampling program again this past year, And what you're seeing here is these different egg count categories down at the bottom. So egg counts are how we base, or what we base our management recommendations on. So the number of eggs in a little bit less than half a cup of soil or 100 cubic centimeters of soil. So here, for example, this bar is fields with between 50 and 200 eggs per 100 cubic centimeters of soil. And here's the number of samples, and then I put the percentage of total samples right above the bars. So you can see that there's still 16.5% of samples that are below the limit of detection for SCN. So this is great because obviously it'd be great to have a field that is not infested, but it's also a good thing to begin sampling so that you can catch those infestations early and begin active management and monitoring. So about 10% of samples were at a very low level. At that level, you could expect no yield loss even when planting a susceptible soybean variety. However, you will definitely increase your population density for the next time you grow soybeans. So 29%, we got up to 2,000 eggs per 100 cc's there. Resistant varieties definitely recommended. The third of our samples here were from 2001 to 10,000 eggs per 100 cc's. At that density, we would expect some yield loss to occur with growing even a resistant soybean variety. 10% of samples were above that, meaning that at those population densities, we'd recommend that folks hold off on planting soybeans until you drop back down to this blue area. One sample really kind of took the cake. This was one field near a town called Olivia in Renville County. 106,500 eggs per 100 cc's. So those rows are never closing. I'm amazed that, this is pure corn and soybean country down there. So we do have problems in the state. We have fewer problems up here, but I think we've gotten lucky. And so please continue to sample and manage accordingly. Here is a QR code. We really would ask that you evaluate our performance here today. We need that sort of information for our funders. We also have a handout here. Volunteer your fields, your wheat fields, for the wheat portion of that scouting program. If you have an interest in that, please see me. And then I have my own contact information is on this card. And then we have an advertisement for a program as well.